So let me discuss now the efforts we have put forth in order to develop a neurovascular unit for in vitro screening of therapeutic agents. So, our rationale for this work is predicated on the fact that CNS therapies largely meet the highest rate of clinical attrition, meaning very few therapies developed for CNS disorders actually make it through the clinic, about 7% to be exact, which is much lower than agents developed for other areas. In fact, about only 45% of all of the compounds that enter phase 3 clinical trials actually make it to the market, which is a lot lower than what we see elsewhere. The rate of attrition for compounds treating neurological disorders is even higher, especially when we're talking about crossing the blood brain barrier. And we hypothesize that this is due to the fact that in the early screening of new chemical entities for these treatments, we use these indirect contact co-culture and triculture models to see how compounds will permeate across the blood-brain barrier, which does not mimic the in vivo physiology. In fact, it's actually rather poor for it. So here, briefly, let me introduce the neurovascular unit. We have the endothelial cells, and in the indirect method, we plate these on top of a filter support, but then we lack putting the parasites and astrocytes, which also provide support and lead to the restrictive nature of the BBB, we put them in physically separated by the filter support. And when we are talking in vivo, we see the endothelial cell, the parasite, the astrocyte here, and we also see neurons that can be plated here. As we are seeing neurodegenerative and neurodevelopmental disorders increasing, like Alzheimer's and autism spectrum of disorders, we really need to do better at improving the clinical efficiency and translation of these therapies in order to mitigate the growing problems with the diseases that are emerging. So what we have done here, and this is a brief schematic, instead of putting these cells on the basement portion of the filter support, we are plating the astrocytes, parasites, and the endothelial cells in direct contact, as evidenced by this transmission electron micrograph that we have here, where we show the endothelial cell, the parasite, and the astrocyte in direct contact as shown or illustrated above. Using this configuration, we've seen that with some marker compounds that we're now limiting the diffusion to about 2 times what we would see in vivo. Whereas the other indirect compound models that have been developed are fivefold higher or greater. We've also optimized this further and added in neurons into the basement chamber. The configuration in direct contact would be too hard in order to do that, so we have just moved them to the basement so we can also separate effects. So what we have developed here is a physiologically relevant model that mimics the in vivo cell culture or the in vivo blood-brain barrier in a direct layered configuration. This enables cell-cell contact, which enhances the restrictive nature of the BBB, which is what we see in vivo. Inclusion of neurons in the basement also allow us to get a little bit higher restrictive properties. So what we see here is if we were to plate the endothelial cells alone, we would see a very high permeability of this 4 kilodalton Fitzy dextran marker. If we were to just co-culture them within a layered configuration of parasites in the endothelial cells or astrocytes, we see a reduction, but we don't see the restrictiveness. When we do the triculture configuration as described, we actually really restrictively limit this. This value is about twofold higher than reported for the in vivo brain. Likewise, we can take compounds like caffeine, carbamazepine, clonazepine that are known to cross the blood-brain barrier, and we have in vivo exposure rank order that was determined in rodents. And we see the rank ordering here when we look at both the triculture and then with the neurons mimics exactly what we see in vivo. So, as such, we have developed this model now, and we have added neurons to allow this neurovascular unit to be formed. And what we're trying to do is to develop collaborations or to work with companies to screen lead chemical entities that they are developing for neurological disorder therapies here. We would also like to maybe work with collaborators who have ideas about ways in which we can modify the application of this to model diseases. We're also looking for potentially investors who may want to license the technology and use it for their own use. We currently have a patent that was recently issued on this, and we have filed several other disclosures to cover the neurovascular unit and other applications that we have been working on in the lab. Thank you for your attention.
Thank you for the opportunity to present our new technology here today. For the next few minutes, I will discuss our new technology for the purification of exosomes from bodily fluids like serum. We call this new device ExoPen. Studies from the past decade have shown that exosomes, which are small membrane-bound vesicles released from most cells, contain biomarkers that can be used for early detection of various cancers, as well as other diseases such as heart disease, infectious diseases, and Alzheimer's. This has resulted in development of approaches to isolate exosome from serum, urine, saliva, as well as other bodily fluids. However, the techniques used suffer from various limitations, including limited purity of the vesicle population, cost of the purification, and ease of use. As indicated here in this slide, there is a significant market for the development of methods to purify exosomes as part of cancer diagnostics. In addition, there is research market, although it's significantly less than the diagnostic market. However, both are growing rapidly with an annual growth rate of about 20%. Our technology for exosome purification is a simple two-step process. The first involves a filtration through a series of membrane filters of different pore sizes to remove larger particles and cells. The second step is to load this material onto a column containing the Captacor 700. The contaminating proteins in the sample are captured in a column, and the exosomes flow through, and these exosomes then can be used for further analysis. Our ExoPen has a number of distinct advantages over other exosome purification methods, as indicated here, including flexibility to use other biological fluids, as well as use in high throughput analysis. The use of exosomes has an additional benefit in that it does not require electricity, which is particularly relevant if you want to use exosomes as part of a diagnostic platform where electricity is limited, such as villages in low and middle income countries where many infectious diseases are endemic. Shown here is a comparison between commonly used purification product ExoQuick and our ExoPen. Exosomes were isolated from 10 different serum samples using either the ExoPen or the ExoQuick. The purified material was spun at high speeds to pellet the exosomes and leave any contaminating protein in solution. The data indicates that the ExoPen removes contaminated proteins in the serum much more efficiently than ExoQuick, resulting in a more purified exosome population. Shown here is a comparison of ExoPen to other commonly used exosome purification kits in the context of speed, ease of use, and purity. As indicated, we believe our product gives highly purified exosomes, but can do so in a rapid and easy method. At present, we are looking for industrial partners interested in developing the ExoPen and for financial support for further testing our ExoPen against laboratory and clinical isolates. We also wish to compare ExoPen to other commercial exosomes purification kits using a diverse sample source. In summary, our data and analysis indicate that ExoPen has a number of advantages over other exosome purification methods and can be developed into a flexible, effective, and scalable method to purify exosomes as a first step in a diagnostic assay. Moreover, the use of exosomes in disease diagnostics is a growing field and will likely continue to grow for the next decade or more. Thank you.
Good morning, my name is Tim Lautenschläger. I'll be presenting on our high sensitivity liquid biopsy technology. I hope by the end of the presentation I've convinced you of its breakthrough potential. Liquid biopsies are currently blood draws that focus on the analysis of foreign biomarkers such as cancer DNA or DNA from a fetus in a pregnant woman. Liquid biopsies are non-invasive and are typically done to avoid a tissue biopsy, which can be uncomfortable, carries a risk of side effects, and sometimes tissue biopsies are not possible if a needle cannot reach the target. The liquid biopsy market is predicted to reach $6 billion by 2023, but that does not include the potential market of using liquid biopsies as a cancer screening tool. The current liquid biopsy technology has limited sensitivity. If you look on the right here at a graph from a recent 6,000 patient circulating cell-free genome atlas paper, I just want to focus your attention here on the common cancers, breast, lung, and prostate cancer. And specifically on the most left is for stage 1. And you see the sensitivity that the blood test detected a stage 1 breast cancer is less than 10%, meaning that out of 10 women with a stage 1 breast cancer, the test only picked up 1 woman as positive and the other 9 women were thought to not have a breast cancer. The numbers with prostate cancer are similar and lung cancer only slightly higher with a 25% sensitivity. That's obviously not good enough. The non-invasive prenatal testing has similar sensitivity issues and is currently not available before week 10 of a pregnancy. So you might ask, why is the sensitivity so limited given that the mutation quantification methods that currently exist have near single molecule capability? So the problem is the biospecimen. If you take 2 tubes of blood for a liquid biopsy test, they might contain a few thousand copies of cell-free DNA. But if you're looking for a mutation that's present in a patient at a frequency of 1 in 10,000, odds are you won't have a molecule of interest in your 2 blood tubes, which means you can't detect it, means your blood test is negative, even though the patient does have that mutation. So how do we overcome this limitation? Well, based on the observation that short DNA fragments from blood can pass the kidneys and are highly abundant in urine, we hypothesized that 24-hour urine circulating tumor DNA testing is the solution to the liquid biopsy sensitivity issue. 24-hour urine tests are widely accepted, and I bet most people know someone who had a 24-hour urine test without any issues. So along the way, we overcame technical challenges that allowed us to develop our IP, such as processing of large urine volumes, retaining short fragments, and the analysis of short fragments. And that's all based here on a graph on the top right that illustrates that in urine, the cancer DNA fragments, so this is an example from a head and neck cancer patient group. If you want to target the cancer DNA in urine, you have to target really, really short DNA fragments. And that's where we developed part of our IP. The overall result of our technology is that we detect over 100 times more cancer DNA pieces than in a 24-hour urine sample than we can detect in a matched blood sample using a standard blood test. And on the bottom right, you just see an example of our urine test where a good response to therapy parallels a decrease in urine cell-free DNA in also a head and neck cancer patient. So what's the market opportunity for this IU technology? Well, the differentiating factor really is the sensitivity that we have. If we detect 100 times more copies than current tests, that will translate into sensitivity gains for various clinical applications. The most interesting is the cancer screening application. We hope that with our sensitivity increase, we can break through that sensitivity barrier that exists that prevents liquid biopsies from establishing a cancer screening market. The current average reimbursement for clinical circulating tumor DNA tests is $2,800, and our technology adds about $200 or so in cost. So we don't expect a huge price difference. The current tests have a 5 to 14 day turnaround time, and our approach adds another 1 to 2 days to whatever routine test you're currently doing. And we can develop many niche products on a shorter time scale. One urgent need exists for stage 4 patients with small cancer volume who currently all have negative test results. But with our test, our 24-hour urine test with more sensitivity, we will be able to identify many patients with targetable mutations, making them eligible for highly effective therapies that without our test would not be able to get these effective therapies. Similar non-invasive prenatal tests, if we manage to establish a test week 5 or 6, that gives a family a lot more time to think about test results than a test that's done week 10 or later. So I hope I convinced you that our technology has potential to be a real game changer. We're looking for investors and grants to establish a sensitivity superior for several clinical indications. We're looking into partnerships with industry or academics to develop market-ready, high-throughput, short fragment sequencing method to get ready to enter the cancer screening market. We're looking for business partners to help us prioritize these opportunities and bring this IAU tech to the market successfully. With that, I want to thank you for listening, and I'm looking forward to talking to you.
My name is Pavlos Vlachos, and I'm Professor of Mechanical Engineering at Purdue University. I'm here to talk to you about Cordiant Technologies and our vision to deliver accurate, affordable, and accessible heart diagnostics for everyone. The market we are focusing is associated with congenital heart disease. That means heart disease because of a birth defect. Every year in the United States, there's 4 million children born, out of which 40,000 will have a congenital heart defect. However, only 1 in 10 pregnancies is going to receive a 30-minute extended screening to assess cardiac health and function. As a result, 50% of congenital heart disease occurs in patients that were not screened and went on undiagnosed and undetected. Critical congenital heart disease hospitalizations had the highest mean and median costs of healthcare for all birth defects considered, and pediatric congenital heart disease hospitalizations accounted for $6.1 billion of hospital costs. This translates to 15% of costs for all pediatric hospitalizations in 2009. Now, what is the total market size here? In the US alone, there's 4 million pregnancies annually and upward of 200 million worldwide. This vulnerable population currently has no access to robust, accurate diagnostic tools for assessing cardiac health. Our technology provides the only comprehensive solution to clinically measure the heart function from the fetal stage in utero to the neonatal and pediatric stages and provide clinical values to inform diagnosis and clinical management. Our tools are capable of analyzing both B-mode and color Doppler echocardiograms, quantify the cardiac chamber and from there the morphology, the cardiac mechanics, quantify cardiac indices, and there calculate flow and hemodynamic parameters. Although commercial systems of echocardiography vendors provide analysis tools, all of those tools to date have been developed for adult patients. As a result, they fail to meet the needs and challenges associated with fetal, neonatal, and pediatric subjects. So currently, there is no commercial solution available to address the needs of this vulnerable population. What we're looking for is for collaborative partnerships and investors in order to translate our technology to the clinics. Thank you.